As we have established in previous units, genetic diversity is a key factor in helping species survive and reproduce, providing certain adaptations that can help a species adjust to changes in the environment. The process that allows this variation is known as meiosis and fits in with Mendel's claims on hereditary traits and variation. In this video, we will display the process of meiosis by detailing each of the steps required for it to occur. First of all, we have the interphase. Rather than being part of meiosis, the interphase is the buildup before the process, where the sex cell gets ready to undergo meiosis. First, the cell undergoes 3 mini-phases in which it grows and creates chromosomes. In the G1 phase, also known as the first gap phase, the cell begins to prepare for cell division and DNA synthesis. The cell accomplishes this by increasing its size and synthesizing proteins, RNAs, and centromeres, the DNA sequence that links 2 sister chromatids. Afterwards, the cell moves into the S phase, where it will continue to grow and produce any necessary enzymes for DNA synthesis. The main event of this phase, however, is the duplication of the DNA, doubling the amount of chromatids by providing each one with a sister chromatid. Note, however, that the amount of chromosomes stays the same, as the amount of centromeres stays constant. In our 2n equals 6 cell, we now have 12 chromatids, but still have 6 chromosomes. This is because the chromosome is defined by the centromere. Finally, in the G2 phase, or the second gap phase, the cell continues to grow while synthesizing necessary proteins. Organelles such as centrioles are also assembled in this phase for future use in meiosis. Now officially starting meiosis, we have prophase 1. During this first prophase, the chromosomes thicken up and form pairs with other chromosomes of similar size and genes, forming homologous pairs. The chromosomes then exchange bits of DNA, which causes them to change and have variations. This process is known as crossing over and turns the chromosomes into recombinant chromosomes.
Metaphase 1. During metaphase 1, the chromosomes will start to move to the middle of the cell. They are in pairs in the middle, and the nuclear membrane which surrounded the chromosomes now disappears. Fibers surround the chromosomes, and the centrosomes are on the opposite sides of the cell. Anaphase 1 is where the chromosomes are starting to be pulled away by the spindle fibers. This separates the pairs. Telophase 1 is now where you have 2 newly formed nuclei and you have 2 new daughter cells. This process of breaking up is called cytokinesis. The 2 cells have 3 chromosomes and 6 chromatids.
At this point, it is important to keep in mind that the 2 daughter cells are now haploid cells, which are cells that contain only a single set of chromosomes. Notice that now the representation of each daughter cell is n equals 3 as opposed to the initial 2n equals 6 because the chromosomes split in half amongst the 2 cells. This contrasts with the initial 2n equals 6 diploid cell at the start, which is a diploid cell because the cell contains both sets of chromosomes, hence the 2 in the 2n equals 6. This leads us into the second part of meiosis, meiosis 2, which just like in meiosis 1, begins with the prophase, namely prophase 2. In prophase 2, the nuclear envelope of both cells dissolves, resulting in the chromosomes to condense similar to how it does in prophase 1. Also, the centrosomes in each cell duplicate and then migrate to opposite ends of the cell. The spindle fibers begin their formation. After prophase 2 comes metaphase 2, where the nuclear membrane surrounding the chromosomes has disappeared and the centrosomes have finished migrating to the opposite poles of the cell. The condensed chromosomes drift to the middle of both cells, the equator, and line up in a single file line. Keep in mind, the spindle fibers keep the centrosomes apart and at the same time attach to the chromosomes at the centromeres. This sets the stage for the next phase, anaphase 2. In anaphase 2, the sister chromatids are separated and are pulled away from each other by the spindle fibers in the cell. This results in the final phase, telophase 2. In telophase 2, the 2 daughter cells divide in 2, making 4 cells in total through a process called cytokinesis. Each of the 4 cells now harbor 3 chromosomes and 3 chromatids. Note that each of the 4 cells will be different from each other because they have different chromosomes that have different alleles. These 4 newly created cells are known as sex cells or gametes, which are the haploid cells that each contain 1 set of chromosomes. Now, how does meiosis connect to Mendel's claims? While we were detailing the process of meiosis, you may have been able to draw some connections to Mendel's claims. In fact, many of Mendel's claims are firmly supported by meiosis. Starting with Mendel's first claim that states alternative versions of inheritance factors account for a variation in inherited characters. This can be observed during prophase 1 as the chromosomes match up in pairs with similar chromosomes crossing over to exchange DNA information resulting in variation. Furthermore, when they are split up in telophase 2 into the 4 gametes, we can see how there are 4 alternate versions of the chromosomes, one in each sex cell, or inheritance factors that result in variation. Moving on to Mendel's second claim that for each character, an organism inherits 2 copies of an inheritance factor, one from each parent, can also be seen in telophase 2, as each gamete only has 1 copy of the chromosome or inheritance factor. This brings us into Mendel's next claim, the law of segregation, that states the 2 alleles for a heritable character segregate during gamete formation. We can observe the law of segregation in anaphase 2 as the sister chromatids are separated by the spindle fibers and later placed in separate gametes during telophase 2, which connects the idea that the alleles which are determined by the chromosomes are separated as a result of the anaphase. Finally, there is Mendel's law of assortment that claims genes assort independently, and we can see this during metaphase 2 because the chromosomes drift to the middle of the cell and line up there independently for assortment in the anaphase. They are then pulled apart and sorted into separate gametes in telophase, displaying how their assortment does not depend on any other genes and happens completely independently.